Hello, this is Professor Torsen. This is the recorded lecture for Chapter 10, Evolution and Natural Selection. So, of course, because this chapter is about evolution, one of the learning objectives is to be able to explain or describe evolution. But this chapter, we're also going to be looking at the evidence for evolution, and there's 5 specific lines of evidence. We are going to look at 4 different mechanisms that can cause or lead to evolution. And we are going to look at one specific kind of evolution called natural selection. So I think many of us, when we hear the word evolution, you might think Darwin. So Charles Darwin did do significant work in the field of evolutionary biology. But there were other people who also were important in this development of the theory of evolution. There were geologists who studied fossils and realized that there were species existing in the fossils that no longer exist, extinct species. There were also geologists who had made discoveries related to the age of the Earth. So I've got a couple highlighted here, but prior to the 1700s or so, many people believed that the Earth was, a couple thousand years old. Many people thought species had been created separately and that they never changed after creation. So this work of these gentlemen here, as well as others I haven't mentioned, really changed the way we think about species and the way species are actually able to evolve over time. So if we take a brief look at the work of Charles Darwin, he traveled around the world on a ship called the HMS Beagle. And part of that trip went to the Galapagos Islands. Now, Darwin took a lot of notes, kept a lot of journals, made tons of drawings on his travels, but he made 2 especially important observations during his time at the Galapagos Islands. And that really had a huge impact on the development of the theory of evolution. These observations were that Darwin saw that different island finches all seemed to resemble each other, as well as resemble the finches found on the mainland. He also saw a similarity between fossils of extinct species and the current species living in the same area. So Darwin published his work in a book called The Origin of Species. And again, this really had a big impact on the way we understood the world. So prior to the work of Darwin and the other people who I didn't have time to mention, but prior to this work, people thought the Earth wasn't that old, a couple thousand years old, and that the Earth and the living organisms on it did not change. However, after the work of Darwin and these other men, the views and the understandings of the world changed drastically. So we learned that the Earth is much older than 6,000 years old. And we also learned that the Earth is not a static entity, that the geology of Earth is dynamic, it's changing, and therefore the living organisms on Earth also change. So we can look at an example of evolution in an experiment presented in your textbook. This particular experiment is using fruit flies. And what they did is they took 5,000 fruit flies and they put them all in a container. And they removed the food from the container and then counted all the dead fruit flies. And so you can see a graph of their results here. And what you notice is that this is time 0 is the point at which the food was removed from the container. So right around 5 hours after the food was removed from the container, we see the first few deaths of fruit flies. So these fruit flies are the first ones to starve to death. And then as time continues, more and more fruit flies die, and then it starts tapering off because at this point, the majority of fruit flies are already dead. And the last fruit flies made it, looks like about 35 hours without food. This graph is called a bell curve, and it's showing you the population. So the population being all of the fruit flies that we started with in that container. And the interesting part of this bell curve is the center, and here's the vertical line showing you where I'm talking about. And this shows you the average amount of time a fruit fly lived without food. So on average, a fruit fly can survive for about 20 hours without food before he starves to death. So the researchers continued this experiment, and what they did is they took another set of 5,000 fruit flies and put it in a container, removed the food, and when 80% of the flies had starved to death, so only 20% of the flies still were living in the container, they put the food back in. So the surviving 20% of the flies were able to eat and then reproduce, and then the scientists were able to look at the next generation. So they took the next generation and repeated this process. So they took the fruit flies, put them in a container, and then withheld food, and then measured the amount of time to starvation. And just by doing this one time through, they saw an increase. So originally, on average, fruit flies lived for 20 hours without food, but after one generation, that average was increased to 23 hours. So on this slide, we're looking at a summary of their results, and they repeated this process for 60 generations. And after 60 generations, they saw a huge increase in the amount of time the fruit flies were able to live and survive without food. So remember, we started at 20 hours average time to starvation. And after that first generation, it increased to 23 hours. Well, 60 generations later, and it increases all the way to 160 hours on average before starvation. So this is an example of evolution. And by evolution, we're defining it as a genetic change in a population. So remember, when we're looking at this data, we're talking about a population of organisms. We're not talking about one particular fruit fly. We're talking about a large population of fruit flies. So as we said, evolution is a change in a population over time with respect to specific traits. So if we combine this with what we learned in previous chapters, we would say this slightly differently. We would say that evolution is a change in the frequencies of alleles present in the population. And one important thing to remember, again, is that we are talking about evolution of populations. We're not talking about evolution of individual organisms. So if we look at this figure here from your textbook, we're looking at tiger fur color. So this pie chart is showing you that most of the tigers in the population are orange, and therefore most of the alleles for the color gene are for orange because most of the tigers are orange. But there are a few white tigers, so there are a few of those alleles for white fur color. So over time, we see this population shift. If we look at this last pie chart, right, we still see that orange fur color, and therefore the alleles for orange fur are still the most common, or the majority of alleles present in the population. But the proportion of those alleles has decreased because we see an increased presence of those alleles for white fur. So we have looked at an example of evolution in fruit flies. And before we continue on this talk of evolution, I want to go over the lines of evidence that support the theory of evolution. So there are 5 primary lines of evidence for evolution, and we're going to go through these one by one. So number 1 is the fossil record. Fossils are the preserved remains of organisms from long time ago, and that can be combined with radiometric dating. So they can use radiometric dating to age the rock and then have a rough estimate of when that organism passed away and became a fossil. The important thing to remember with the fossil evidence is that there is some limitations to it. So only certain physical structures can leave fossils. And the second thing to remember is that fossils, they require a very specific set of conditions in order to form. And so if you look at an example from your textbook here showing you the evolutionary history of horses. So here we have the modern day horse, and all of these are extinct species here, and it is known, or these species rather, are known because of the fossil evidence. The second line of evidence that supports evolution is biogeography, which is the geographical distribution of living organisms around the world. So this is essentially what Darwin was using. He noticed similarities, common structures among species living close together, but on different islands in slightly different habitats. The third line of evidence for evolution is comparative anatomy and embryology. So embryology is the development of the embryo, and they can look at these embryological stages and look for similarities in the development of the embryo. In terms of comparative anatomy, you can see an example from your textbook here, looking at the structure of our arm and the bones contained in our arm. And those bones are very similar to the bones in the front leg of a horse, in a bat wing, and in the fin of a porpoise. So because there is a similarity in these bones are what we call homologous structures. So structures of the body that are very similar in different species, but they may serve different functions, right? So our arms are used for many things, But for a horse, that forelimb is specifically for walking. And the bat forelimb, of course, is for flying. This is an important distinction between homologous structures and analogous structures. So again, homologous structures are very similar structures, but they may have different functions. Homologous structures suggest common ancestry. So they suggest that these organisms are related evolutionarily speaking. Analogous structures, though, are kind of the opposite. So analogous structures are structures that serve similar functions, but they don't have a similar structure. So for example, here's a dragonfly and here's a bat. Both of them have wings that enable them to fly, but the structure of the dragonfly's wings and the bat wings are very, very different. So even though they serve the same function, the structures themselves are quite. The fourth line of evidence for evolution is molecular biology. Molecular biology allows us to compare the genetic code, or DNA, of individual organisms. We can compare across species and look to see how similar or how different the DNA sequences are. The fifth and final line of evidence for evolution are experiments performed either out in the field or in the lab. So an example of a laboratory study would be the fruit fly experiment we discussed earlier this chapter. Another example of a laboratory study is looking at the development of antibiotic resistance in certain bacteria called Staphylococcus aureus. And an example of a field study is shown here in figure 1043, in which people investigated the grass in different areas of a golf course. There are 4 mechanisms by which evolution can occur. Remember, evolution is a change in the frequencies of alleles within a population. So we're going to look at the 4 mechanisms behind this change in the upcoming slides. So the first mechanism of evolution we're going to look at is mutation. And we've learned this term already. Mutation is an alteration of the base pair sequence in DNA. So mutation is the ultimate source of genetic variation in a population. Mutations can be caused by any number of factors, things like radiation or various chemicals can do it, and it can also just happen randomly by accident due to errors in cell machinery. So because mutations are random, we can't predict which individuals will have a mutation, and we can't predict what the effect or the consequences of a mutation is. Keep in mind that only mutations that affect gametes, the reproductive cells, are able to be passed to offspring. So mutations in somatic cells won't be passed on to your offspring. The second mechanism of evolution is genetic drift. In genetic drift, we see a change in the frequency of alleles in a population, and this is especially impactful in smaller populations. So for example, here we see at the bottom of figure 1012, we might see this pie chart in which there is slightly more of this teal color compared to the green color, and these represent 2 different alleles. Over time, we can see a drift such that there are more and more of the green alleles and fewer of the turquoise alleles. There are two kinds of genetic drift. The first is called founder effect. In founder effect, a small number of individuals leave a population and found a new isolated population. So for example, if we're looking at this pie chart again, we are looking at two different alleles. One is being represented by this blue color. The other is being represented by this green color. So we're looking at a pie chart in which most of the alleles present in the population are this green color here, which is representing the dominant allele. Let's say a portion of this population breaks away and moves to a new location. So we're taking a slice of the pie out. And we happen to slice the pie right here. And when we look at this separate population and we look at the proportion or the frequencies of alleles in this subpopulation from the main population, we see that the frequencies are different. Green, which is representing that dominant allele, is still the majority. However, we see a larger percentage of that recessive allele in this newly founded population compared to the original source population. That essentially is affected by where you take a slice of this pie. The second type of genetic drift is called population bottleneck. So there is some event that happens. It can be a big change in weather, a big change in the environment, or any other extreme events. And the extreme event then kills the majority of the population. And so we're reducing the population size by a very significant amount so that we have a small number of individuals compared to the original population. This is represented in figure 1014. Here we have a pie chart representing the original source population. And you can see there are 4 different colors here in this pie chart representing 4 different alleles. So if there is a large extreme event, big change in weather, what have you, that can cause the demise of a significant number of individuals in this population. So after that large event, we see that the only remaining individuals here are the blue and the green alleles. And so that will form a new population. And now we've reduced the number of alleles that we had originally in the source population. So the purple and the yellow alleles are no longer present. And we've also changed the frequencies of those alleles, right? So before the extreme event, we had roughly the same amount of blue and green alleles, but now in the new population, the remaining population after that extreme event occurred, we have mostly blue alleles and a few green. The third mechanism of evolution is migration, also called gene flow. Migration is when some individuals move from one population to another. So for example, here we are looking at 2 distinct populations living on opposite sides of this mountain range here in the center. So both population 1 and population 2 have the same type of alleles being represented by green and then this kind of turquoise blue color. But the population here on the left-hand side, population 1, is got a different frequency of those alleles compared to population 2. You can see population 2, what is maybe 95% turquoise, whereas population 1 is maybe 70% turquoise. So if there is a migration or gene flow, as pictured here, part of this population on the left-hand side moves over to the right-hand side. And the result is that we are changing the allele frequencies in the resulting populations. Again, because of how the population was sampled and the members of this population that migrated over the mountains happened to contain a very high proportion of the green allele and very few of that blue or turquoise allele. The fourth mechanism of evolution is natural selection. Natural selection is likely what you normally think of when you think of evolution, but again, it's not the only mechanism of evolution. For natural selection to occur, 3 conditions must be met. Number 1, there must be variation for a given trait within a population. Number 2, that variation must be heritable. So that variation must be able to be transmitted to the next generation. And the last condition is called differential reproductive success. So this is a fancy way of saying some individuals reproduce better than others. So if we take a closer look at differential reproductive success, essentially what is happening is that more organisms are born than can survive given the resources in the environment. So organisms are continually fighting for access to those resources. And therefore, some organisms are going to be more likely to survive and reproduce, while some organisms just won't be able to get access to those resources and won't be healthy enough to reproduce. Here we can see an example of natural selection occurring here in nature in figure 1020. So again, the 3 requirements for natural selection is that there is variation within a trait. For example, there might be 3 different alleles for a gene that determines how fast these rabbits can go. So there's natural variation in this trait. The blue rabbit is the fastest, the green is medium, and the yellow is the slowest. This trait is heritable. So the trait of running speed is transmitted from parent to offspring. So slower rabbits have slower babies, medium speed rabbits have medium speed babies, and those fastest rabbits have fast babies. And this can lead to differential reproductive success. So you can imagine that out in the world, rabbits have a lot of predators, and here we see a fox. So the fox will be able to eat the slow rabbits and the medium rabbits, but those faster rabbits are fast enough to escape the fox. And so these faster rabbits are able to survive and reproduce, while these slower rabbits are not. When learning about natural selection, you may have heard the phrase survival of the fittest. What this phrase is really saying is that it's the individuals within a population who are best suited for a particular habitat that are going to be the ones who have the greatest reproductive output. So if we consider mice living in two different habitats. So we're talking about the same species of mouse. Here in this lighter colored habitat, the lighter colored mouse is better suited for that environment. The slightly darker colored mouse is more easily seen from the background. So in this particular environment, survival of the fittest means that this lighter colored mouse is better suited to the environment and will have greater reproductive success. But if we look at the same species of mice in a different environment, an environment where it's a little bit darker, now suddenly that darker mouse is better suited to the environment. And this darker mouse is going to be the one that will have greater reproductive output. One more thing I want to mention about natural selection is that natural selection does not lead to perfect organisms. There are several factors that prevent populations from regressing to some level of perfection, whatever that may be. Those factors include the environment changing quickly. So environment can change much more quickly than a population can adapt. Another factor is that variation is needed as the raw material for selection. Again, there needs to be variation for a trait in order for natural selection to occur. If everybody has the same trait, there's no variation, no one will have a greater reproductive success than another. The last factor that can prevent populations becoming perfect organisms is that while there might be variation for a trait, there might be multiple different alleles for a given trait. If each allele allows the organism to be suited well to the environment, to the point that it doesn't matter what phenotype or what alleles for a trait that you have, you are still suited well to the environment, and there's no difference in the reproductive success of those different phenotypes, then essentially all phenotypes have the same fitness, and there won't be any selective pressures to select any particular phenotype. Humans have been using artificial selection to breed plant and animal species. This has been going on for thousands of years. So when humans breed for a specific trait, a desired trait, it is called artificial selection. So one example of this was, again, that fruit fly experiment from earlier in the chapter. This was artificial selection. The researchers were selecting the fruit flies that could survive the longest without food, and were selectively breeding those. And we saw that over the course of 60 generations, the average time to starvation increased drastically from 20 hours to, if I remember correctly, 160 hours. So there are three different kinds of selection, and for all of them, we are starting with a uniform population. So in gray here is the normal distribution, this nice bell curve here, where we're looking at the number of individuals with a particular phenotype. So in this example, we're looking at the size of breast muscles in turkeys. So before any selective breeding, there were on average turkeys with this size breast muscle, and then there were some with smaller and some with larger. But if farmers always selected the larger turkeys to breed over many generations, we will see a shift in the population to this turquoise color. So we still have a range of turkey breast size, right? We still have smaller, we still have larger, but when we compare this population to the previous population, we see this shift towards the right-hand side. So the average turkey breast is much larger now than it was prior to that selective breeding or artificial selection. That is called directional selection. Here in the middle, we're looking at stabilizing selection. And in this example, we are looking at birth weight of human babies. Again, we have gray representing the population before any selective pressures. So there is a range in birth weight, and there are some babies who are born very light, some babies who are born heavier, and the average baby size is right around 7 to 8 pounds. So after various advancements in medicine to deal with various complications in pregnancy and birth, we have seen a reduction in the very light babies and the heavier babies. And so here in this turquoise blue color, we see the new distribution of human birth weight. So we still have a range. We still have slightly smaller babies, medium-sized babies, and larger babies, but that range is much more reduced. So we've gotten rid, or we're losing some of these extremes. The opposite of stabilizing selection is disruptive selection. So in stabilizing selection, the selective pressures meant that an intermediate phenotype was the best. We got rid of the extremes. In disruptive selection, it's the opposite. So the intermediate phenotype is least fit for the environment, and the extreme here on either end are better fit. And the example here is coho salmon. Again, in gray represents the original population and the distribution of sizes. But after selective pressures, those medium-sized fish are far fewer because of this disruptive selection. That concludes the recorded lecture for this chapter. Thanks for watching, and remember, go wash your hands.